Hi everyone, welcome back to chapter 7 from DNA to protein. In part 2, we're going to go ahead and take a look at what translation will bring to the process. So by now we have conquered part 1, so we are all experts on transcription. We realize that it takes place in the nucleus of a eukaryotic cell, and that the whole goal of transcription is through the 3 steps of initiation, elongation, and termination to ultimately take a gene from DNA and transcribe it into an RNA molecule. You also want to keep in mind that there are different types of RNA, and we talked about the 3 most common: the messenger RNA, the tRNA, the transfer RNA, and the ribosomal RNA, also known as the rRNA. Also keep in mind that for messenger RNA, towards the end of part 1, we discussed some of the modifications that needed to be done to make sure that the molecule was nice and stable and was able to be successfully exported out of the nucleus. So we added the cap onto the 5' end, we added a poly A tail onto the 3', and then we did our splicing, which allowed us to get rid of the introns and put the exons together, because the exons are the ones that are expressed and are the sections that will be exiting the nucleus into the cytoplasm. And where we are now currently located will be within that particular region, so that we can go ahead and complete the next step of decoding our DNA, and that will be translation. Now, the definition of translation is converting RNA into proteins, and it's going to utilize all 3 of the RNAs that I've mentioned to you before. Your mRNA will be your messenger RNA, and that will probably be the most important one, because as it indicates in the name, it will be carrying the specific protein directions that need to be synthesized. The rRNA will work together with ribosomes, hence the name ribosomal RNA, and it will be doing the reading of that particular code. And then the tRNA, the transfer RNA, will be the one that will be bringing over the amino acids, which obviously are the monomers, or the building block of the polypeptide, the protein chain that we are trying to produce. So let's go ahead and let's start taking a look at mRNA. mRNA, as I said to you, is our messenger RNA, because it turns out that once you have made your complementary match of RNA based on your template DNA, what we're going to do in the translation step is we're going to take our mRNA molecule and we're going to go ahead and break down the nucleotides in sections of 3. And each section of 3 nucleotide is then going to be called a codon. And this is going to be important because it turns out that the codons will form a reading frame for the rRNA to comprehend, and as it's reading the codons in sets of 3, it will correspond with a particular amino acid that the transfer RNA will be bringing over. Now, it turns out that with our 3 nucleotides separated, there can be a combination of 64 different codons. In that combination of 64 codons, we will also include some stop codons, and we will definitely also have a start codon. In a little bit, I'll show you an example of a codon table, but for now, just keep in mind that when you hear your mRNA, when it comes to the translation, please keep in mind that the way you're going to read that molecule is going to be by reading its codons, and each codon is represented by 3 nucleotides. Now, the illustration that's right over here on your right hand side is showing you what we call reading frames, and it's showing you that within one strand of mRNA we can go ahead and we can come up with different ways to read our codons. It doesn't necessarily mean that, like in example 1, you start with the first 3. You can also kind of start with the second one, or you can start your codon after the third one. There's different ways to kind of break it down into codons. But it turns out that within those codons, there is one that will indicate the official start of translation, and that is obviously where your start codon will come in, AUG, and the end of translation will be signaled by reaching one of the stop codons. So there will be a clear codon to indicate where we're going to start, and there will be a clear codon to indicate when we will stop our translation and our production of our polypeptide chain. Let's go ahead and take a look at our codon table. So here's an example of a codon table. Now, disclaimer, I borrowed this codon table from another McGraw-Hill book. I did not use the one that was in yours, only because I think this one is a little easier to read. But by all means, all codons tables will be set up in a very similar way. They will allow you to look for all the different combinations of the four nucleotides paired in 3. So as you look in the boxes, you're going to see that they're filled with U, C, A, and G, your uracil, your cytosine, your adenine, and guanine, because those are the 4 nucleotides that we are familiar with when it comes to RNA. And within the little blue boxes, you can see that they have come up with all the different combinations that we can form our codons with those 4 particular letters. And within that, you're going to notice that they will code for the different amino acids. Now, there are 20 amino acids, and there are 64 possible combinations. So it's quite common to have multiple codons that will give rise or that will code for the same exact amino acids. And you can see that in your table, that you have brackets that will all lead you to the same amino acids. Now, the easiest way to read this particular codon table is to start on your left-hand side, where it says the first letter of the codon. And then what you'll do is, once you've identified your first letter, you will move across the table, looking at the yellow boxes to identify your second letter of the codon. And once you've come across that, then it's easy to find the third option, which will then allow you to successfully identify the amino acids. So let me, for instance, give you an example. Let's say you come across a question and it says, which amino acid does the codon AAG represent? Okay, so the question is, which amino acid does the codon AAG represent? You can scavenge through the table if you want, but go ahead and follow the rules of, or follow the suggestions of the first and the second codon. So go ahead and find your A on the side, on the left-hand side. So here's my A. Once I have that, I'm going to go across, and looking on top, I'm going to find my other A box, which is my third category right here. Right, here's my other A. And then within that box, I'm going to look for my G, and it's right over here. AAG, and I can see that this particular codon is going to produce lysine for me. Same thing can be done, like let's say, for instance, if we're going to do another codon, let's go ahead and come up with UCA. UCA. So start on the side, find your U, go across, find your C. There we go. That brings us to this box, and UCA will bring us and produce serine. There you go. So as you can see, in this case, all 4 combinations of the codon, whether you had UCU, UCC, UCA, or UCG, would be able to produce the same amino acid. And your body does this for a specific reason. Remember in part 1 how I noted to you that the RNA polymerase isn't as good at proofreading and correcting its mistakes? Well, your codon table has been built in to allow for a little mistake, but to still get ultimately the correct amino acids. And the way it works is that, as you're going to notice, a lot of times, if you get the first and the second letter correct, then the third letter is often in what we call a wobble position. That means that you can kind of mismatch the third letter. It doesn't have to be the correct letter, but it will still often produce the same amino acids that you require. So in the case of serine, if there was a mutation within the third letter of the codon, and for instance, the U was replaced with a C, or the G was replaced with an A, it wouldn't really matter. As long as the U was the first letter and the C was the second letter, we would end up with the same exact amino acid. So this is an excellent way of neutralizing some of the mutations that you might come across by offering multiple options for the third letter, thereby guaranteeing that you most likely will still get the same amino acid. I also want to point out, and you can find this color-coded in your table, that there are 3 stop codons that have been identified: UAA, UAG, and UGA. These are your 3 stop codons. Notice how the stop codons do not produce an amino acid. They are going to go ahead and cause release factors to be introduced into the system, and that will eventually halt the whole translation mechanism. We're going to talk more about that in a little bit. You also have one start codon, which you can find on your left-hand side. It is marked by AUG. AUG is a universal start codon. All organisms that do translation will start their translation by finding those three letters, AUG. It will also code for the amino acids methionine, but oftentimes that amino acid will be cleaved before the protein is folded in its final form. But either way, AUG is going to be your start codon. You do not have to memorize the codon table for your exam. In fact, I will be providing you the codon table so you can utilize it as you're conducting some of the questions. The only difference in the codon table that I will provide versus the one that you're looking at is that in my codon table that I'll give you for the exam, I am going to remove the start terminology because that is the one codon I want you to memorize. I want you to know that AUG is going to be your start codon. So I'm going to remove the word start from our codon table because definitely I'm going to ask you what's the start codon, and I want you to know it's AUG without having to reference to the table, whereas I'm not going to change anything else. So you don't have to memorize the stop codons. You don't have to memorize which one's going to give you proline or valine. You will have access to this table as you are conducting your questions. Alrighty, so we're going to explore the codon table a little bit more towards the end of our chapter because I will be providing you with a DNA sequence that we're going to work our way through and produce RNA and protein. So we're going to do transcription and translation and see what our final polypeptide chain will bring us. Let's go ahead and take a look at our next RNA molecule, and this one's going to be tRNA. tRNA is going to be our transfer RNA molecule, and this molecule has a very particular 3D shape. It is often compared to a clover leaf. That's why that little drawing is right over here. And as you can see in the 3D rendition of your tRNA, you're going to have some complementary bonding between the guanines and the cytosine, the adenines and the uracil. Notice the little red lines indicating the hydrogen bonds that are holding these complementary sequences together. But probably the most important thing about the tRNA are going to be what you find in its 3' end and its area that's indicated as an anticodon loop. The 3' end is very important because the transfer RNA is going to be responsible for moving around and bringing over the required amino acids that each codon is specific for. And those amino acids are going to be carried on the 3' end. So this little green ball is representing the particular amino acid that this tRNA is carrying. Now, how does the tRNA know where to drop off the amino acid? Well, it turns out that it will have an anticodon section that will complementary match with the codon for its particular amino acid. So as the rRNA is reading the codons, it's communicating with the tRNA to bring over the amino acids. When the amino acids show up, obviously we have to put it in a certain order to produce our overall desired protein. So what we're going to see happening is that the tRNA will complementary match its anticodon to the codon on the mRNA to guarantee that the amino acid is dropped off properly. So for instance, in this example, the anticodon is GAA. So what would be the codon that it would complementary match to? Well, remember your base pairing rule. So if there's a G on one side, there will be a C on the other end. And if there is an A on one side, there will be a U on the other end. Remember, we're doing RNA, so no T's. So that means that my codon on mRNA is CUU. It is coding for my family amino acid. And when that amino acid is brought over, we're going to temporarily, really quickly, match the anticodon to the codon to guarantee that it's being placed in the correct spot. Now, the next question is, how does the tRNA know which amino acid has to go on its 3' end. How does it know which anticodon matches the amino acid, right? So we know the anticodon will match the codon, and we're going to drop off the amino acid. But how did we initially get that amino acid on the proper tRNA end so that it matches perfectly with the anticodon and the codon? Well, in order to do that, we're going to rely on a very particular enzyme called the aminoacetyl tRNA synthase, which we're going to learn a little bit more on our next slide. All right, so here's our aminoacetyl tRNA synthase. Try to say that 5 times fast, right? Me and my lisp. My tongue almost fell out. Anyways, so what we're looking at here is this beautiful protein enzyme complex. And this particular synthase enzyme is going to make sure that the proper amino acid is linked to the anticodon, so it's going to be covalently coupling the correct amino acid to the proper tRNA. Now, it turns out that within the cell, you will have a different synthase for each amino acid. So in general, you'll have 20 of these because there are 20 amino acids that we have to play with. Now, the way it's going to work is, as you can take a look at your illustration on the side, in blue, we are representing our tRNA, and then here in this green structure, you can see that this is your synthase enzyme. It's making multiple contact points with the tRNA, including interacting with its anticodon loop and its amino acid accepting arm. And it will be going ahead and attaching the amino acid that is properly matched to the anticodon to the 3' end of the tRNA. And it turns out that this reaction of linking together the amino acid on the 3' end will create a very high energy bond, and it will allow the tRNA to then be called a charged tRNA. And this charged tRNA will allow for the energy in this high bond created by holding on to the amino acid, its high energy bond. It will allow that energy to be used later on to link the amino acid to the growing polypeptide chain that it will encounter when it drops off its amino acid to the rRNA and the mRNA. So your aminoacetyl tRNA synthase is going to be your enzyme complex that guarantees that the proper amino acid is bound to the 3' end based on the anticodon that is being carried on each tRNA. Now, once that's done, obviously, then we're going to also rely on the anticodon to make sure that the amino acid is placed in the correct location based on complementary matching the anticodon to the codon on the mRNA. So to guarantee accuracy, it is essential that the synthase enzyme as well as the anticodon and the codon matching are on point to make sure that the amino acid is not only properly transferred, but also properly arranged to produce your end protein. The next RNA is going to be our rRNA. This one is what I like to call our reading RNA. rRNA is basically a combination of RNA as well as proteins. Usually the entire complex by weight, 2/3 of it will be composed of proteins. 2/3 of it will be composed of RNA, and about 1/3 of it is proteins. It is a very large complex. It is very unique in its overall shape, because when we look at it, we're going to notice that it's actually a combination of 4 RNAs mixing together with about 80 small proteins. In fact, please do me a favor and take a look at your illustration that's on your left-hand side. And what it's trying to show you is that your rRNA in its final shape will be composed of a large subunit and a small subunit. Now, we are looking at a eukaryotic rRNA, but the prokaryotic one is very similar in its shape, and it will also have a combination of RNA and proteins within its final composition. Now, if you look on top of the illustration, it's going to go ahead and show you how the large subunit is done by combining 3 rRNA molecules with about 49 smaller proteins and linking them together. Because your RNA is going to be the larger entity and the heavier based on weight, it will ultimately control the overall shape of the subunit. Same thing can be done for the small subunit. The small subunit is composed of one RNA molecule with about 33 different proteins linked together. You will need both the small and the large subunit of the rRNA to click together, and when they click together, they will be able to read the codon sites on the mRNA. In fact, what we can do is we can come over to our right-hand side, and you can see our little 3D rendering of the rRNA. Notice how they've used the lighter green to represent the large subunit and the darker green for the small subunit. And come over here to the illustrated example. You can see them click together. Here in the black dotted line, it is showing you how the mRNA will be feeding through the small ribosomal unit, and it will be reading its codons, so 3 letters at a time. And within its interaction between the small and the large ribosomal subunits, we're going to see 3 different binding sites. We're going to have an A site, a P site, and an E site. And these sites are going to be essential for the tRNA to bring over the desired amino acid and also link that amino acid to the growing polypeptide strain, all while checking its anticodon to the particular codon that is currently being read by the rRNA. Let's go ahead and take a look at all this in motion. So we'll explore a little bit more between the differences between the A site, the P site, and the E site. All right, so here is translation in its multiple steps. And I should probably tell you that very similar to transcription, there are 3 main events that you're looking for. So in transcription, we had initiation, we had elongation, and we had termination, right? All about attaching the RNA polymerase, finding your starting point, matching the template with the DNA to the complementary RNA strand, and then reaching the terminator sequence. Done. In translation, you will also have 3 main events, and those 3 main events are called initiation, elongation, and termination. Yes, ladies and gents, the events will have the same exact names, but obviously what's actually occurring within the steps is going to be completely different, because remember, the goal of translation is to convert your mRNA into a protein strand. So you're going to have different players and different events that are going to be needed to get that done. So let's walk through our steps, and then I'll tell you exactly where each of these events fall into initiation, elongation, or termination, okay? So we'll pick up on that in just a little bit more. But for now, let's go back to our discussion where I said to you that the rRNA is going to be quote-unquote reading the mRNA, and the rRNA is composed of a small subunit, right over here, and this is my large subunit. Now notice how the reading frame of the mRNA fits perfectly within the active sites, right? We have an A, we have a P, we have an E, there we go. So the first thing that we're going to see happening is that the tRNA with its amino acid is going to enter through the A site. So that's the first site that the tRNA is going to encounter. Now remember, we're calling it a charged tRNA because it was successfully able to link its amino acid utilizing the synthase enzyme to the anticodon found in the tRNA. Once it enters into the A site, what we're going to see happening is that it temporarily binds and complementary matches its amino acid, its anticodon with the codon for mRNA. And it will also allow for the amino acid that it brought over, right over here, to form its peptide bond with its neighboring amino acid, which is part of the growing polypeptide chain. So go ahead and take a look at step number 1. Identify your growing polypeptide chain. And as you can see, you have 1, 2, 3 amino acids attached to it thus far. So when a newly charged tRNA comes in with amino acid number 4, we see that in step number 2, while that tRNA is in slot A, it will allow the amino acid to make a polypeptide bond with the previous amino acid that was being held by the second to last tRNA to come into the process, which is being held in slot P. Okay, so P is the tRNA that came before that. Notice how once the polypeptide bond is made, the tRNA in slide P will completely release its amino acid. As this is happening, we are basically done with this tRNA. So how do we get rid of it? Well, we see that the large subunit of the rRNA will shift its frame, follow the arrow, and when it shifts its frame, it will allow the old tRNA that was previously in P to be moved over to the E site. And that is basically a few more milliseconds before it actually can be ejected out of the entire ribosomal subunits. And then what we see happening is once the ejected tRNA is out, it will also allow for the newly charged tRNA, the one that was bringing over the fourth amino acid, to migrate from the A spot to the P spot. And now you can see it's still holding on to its amino acid because it's waiting for the next newly charged tRNA to stop on by. So this one's bringing over amino acid number 5. That will enter to site A. And then the next step will be to form a peptide bond between 4 and 5, so the fourth and the fifth amino acid, so that the large subunit of rRNA can once again slide on over. And as it's sliding on over, it will allow for the tRNA to go over to the E site and then be ejected out. And the sliding mechanism of the large subunit is then closely followed by the small subunit as they're migrating starting from the 5' end of the mRNA as they're migrating towards the 3' end of the mRNA. So that is one of the reasons why when you're taking a look at your rRNA molecule, you're going to see 3 different active sites because each of them will have a particular role. And the formation of the peptide bond in step number 2, that is a perfect example of why your rRNA can also be classified as a ribosome because of the fact that it had its catalytic activity. It's allowing the peptide bond to be formed, thereby catalyzing a new reaction. So sometimes when you read about rRNA, they'll talk about the example that it's a ribosome. And that is because of its ability to do the catalytic action of forming the peptide bond between the newly inserted amino acids. Also notice how even though it has 3 active sites between the small and the large subunit of the rRNA, at all times it's only 2 sites that are occupied. So there's always one that sits empty. So if you look at step number 1, you notice that the E site is empty. Step number 2, still E. Step number 3, the A site. Step number 4, the A site. And then step number 1, once again, the E site. So most of the time, there's only 2 sites that are occupied as we are adding our amino acids to the growing chain, and the tRNA enters through A, goes to P, and then exits through E. Now, I mentioned to you before that very similar to transcription, there's going to be a start and a stop mechanism. And in fact, when we were looking at our codon table, we talked about the fact that there's a universal start codon, and that one is AUG. I remember it as the month of August. That's really, honestly, how I've always remembered it. Now, AUG will be the initial amino acid that will start off translation. So what we see happening is, and you can go ahead and follow along in the figure, this is figure 7-39, is that the rRNA will initially only utilize its small subunit, and its small subunit will be able to recognize the 5' end of mRNA due to the appearance of the cap. It will attach itself to the 5' end, and it will start sliding over through the codon reading frames until it gets to AUG. Now, at that point, what we see happening is that a combination of the small ribosomal rRNA, as well as what we call the initiator tRNA. This is the tRNA that's going to be able to interact with AUG. Notice how it has the methionine amino acid attached to its 3' end. It will be able to recognize the AUG site, and when it recognizes it, it will then go ahead and send a signal over to recruit over the large ribosomal subunit. Now, it doesn't do this by itself because, as you can see from the illustration, in addition to having the small ribosomal subunit and the initiator tRNA, which can recognize your AUG, you will also have the help of other small proteins called translation initiation factors. And these will help guide over the small subunit of the rRNA over to your AUG. The initiator tRNA is unique in many different ways. In addition to the fact that it can go ahead and bind with the start codon, AUG, it is also the only tRNA that can tightly interact with the rRNA molecule and the mRNA molecule without the use of the large subunit. So it's fully able to attach to the codons and read and interact with the codons without the clicking mechanism of the 3 binding sites, the A, the P, and the E. So that's very unique to the initiator tRNA, which can recognize the start codon. Either way, if you come on over to the second part of the illustration, you can see that once the AUG, the start codon, has been identified, we can get rid of our translation initiating factors, and when that happens, it will recruit over the large ribosomal subunit, and it's almost like a clicking action. The small and the large subunit will go ahead and bind together, and once they bind, we will go ahead and see the formation of our A, our P, and our E active site, which means that we can start recruiting over other charged tRNAs to enter and start covalently binding their amino acids so that we can start building our peptide chain. And then it goes back into the same order of it enters through A, it moves to P, and then it's ejected after it goes into E. Now, last slide I said to you 3 main events. We have initiation, we have elongation, and we have termination. And we also have that in translation, just like we have it in transcription, right? So the initiation event in translation is all about finding AUG. So from the minute the small ribosomal subunit identifies the 5' cap of mRNA, that's going to be the start of your initiation. As the initiator tRNA is gliding over and finding AUG, you're still in the initiation step. And it's only after the large ribosomal subunit connects with the small subunit and the whole rRNA is fully formed that you have now reached the end of your initiation step, and you will go ahead into your elongation step. The elongation step is when you're actually matching your codons with your anticodons and you start building your amino acids chain. So the actual work of translation, entering through the A, going to the P, and then the E site, that is going to be part of elongation. So this is when you're growing your polypeptide chain. There's only one more event, and that will be termination. Termination means that you have reached the end, so this is going to involve the fact that eventually the rRNA subunits are going to encounter a stop codon. So let's go ahead and click over to the next slide and see what's going to happen over there. All right, so as you can see on my illustration, we are gliding along, started at the 5' end of mRNA. We're moving over in the direction of the 3' end, and we are reading in sets of 3, so we're doing our little codons. And now what I see happening is eventually the rRNA will come across a codon that's a stop codon. Now remember, stop codons do not signal any amino acids. They don't represent any amino acids. So instead, what you're going to see on your illustration is that entering into the A side will not be another charged tRNA, but instead it will go ahead and recruit over a release factor. the release factor will form almost like a blockade within the A site. And when this is detected, it will go ahead and cause the immediate termination of translation. So this is the final event that needs to occur. And part of that is because the appearance of the release factor will allow the binding of a water molecule instead of another amino acid molecule. And because the water is introduced, it will allow the polypeptide chain to be released from the entire rRNA complex. So there is that little protein that you've been working on. It's released, and when that becomes released, we see that the large subunit of the rRNA, when it shifts like it normally would, it will actually become dissociated, meaning it will fall apart. And when the large piece falls apart, so does the small subunit. And along the way, it will get rid of the release factor and any of the tRNAs that remained in the active sites. So the final step of translation, the termination, means that your rRNA reaches a stop codon. It recruits over a release factor. The release factor terminates the translation sequence. You release your polypeptide chain, and then the rRNA will fall apart, so the large subunit dissociates from the small subunit, and you have officially ended your entire interaction between the tRNA, the rRNA, and the mRNA. So now you have your freshly produced polypeptide chain. So I just want to remind you, and I know you have all become experts on proteins because of our previous chapters, but remember with proteins, shape equals function. So once you have completed your translation, there are some modifications that need to be done to ensure that the protein will reach its full ultimate shape and be able to perform its desired function. So this little illustration right here just reminds you that in addition to having your polypeptide chain, you will also rely on things like your chaperone proteins, different cofactors that will allow non-covalent interactions to occur so that they can go ahead and be upgraded from a primary to a secondary, to a tertiary, to a quaternary structure. And along the way, you will have some different modifications. Remember how we talked about phosphorylation, for instance, as well as that it can start non-covalently binding with other molecules, creating a larger subunit and thereby a mature ultimate product. We should also talk about the fact that your body or your cells have to be able to regulate the amount of total protein that is floating around and that's being used. So very similar to how you can have gene regulation, you can also control the amount of protein. And this can be due to the fact that you can have, for instance, a protein that has a relatively short lifespan. It could also be for the fact that the protein might just be old or misfolded and it's no longer functioning the way you would like it. So we don't want to have any type of disease models pop up due to unwanted proteins hanging around. So there should be a way for us to control the protein breakdown. So in order to do that, we are going to go ahead and utilize these particular proteins called proteases. And these are going to be specialized enzymes that can break down the proteins that are no longer needed or wanted within the body. And what we see happening is that you will create these structures called proteasomes. Proteasomes are going to be these large complexes that you find within the cytoplasm. You can also find them in the nucleuses and nuclei. But a proteasome is this very large complex that will often have in the center of it, it will have a little cylinder shape. And housed in the cylinder, you will put your protease enzymes, so the ones that can break down the proteins. And the way they break down the proteins is by cleaving the peptide bond. So they basically chop it up into smaller amino acids. They will put these proteases within the cylinder section with their active sites showing to the opening or the lumen of the cylinder. So here in red, we see the little active sites. In the meanwhile, in the rest of the cell, you have these other very tiny little proteins that are called ubiquitin, ubiquitin industries right here in red. Ubiquitin is going to go ahead and be able to tag the target protein that you're looking for. So the one you want to get rid of, whether it has reached its lifespan, it's old, it's damaged, or you no longer need it. And it will guide it over to a binding site within the proteasome. Once it's in the binding site, as you can see here in step number 2, it will allow the ubiquitin to become separated from the tagged protein because we want to recycle it all over again. And then the tagged protein will be forced into the cylinder section where it will interact with the active sites of the proteases. And as you can see, the peptide bonds are being cleaved, and what comes out of the proteasome are going to be the individual amino acids that we can then recycle as we continue to complete our transcription and translation step over and over again. So if you think about it right now as you're listening to my lispy voice, your cells are actually doing this. They're doing transcription, they're doing translation, they're modifying the proteins that they're producing, they're breaking down the ones they no longer want to use so that we can recycle those amino acids for another turn around the central dogma of the transcription and translation. And in our final slide, I just wanted to kind of show you all the different steps that we have worked through in chapter 7 as we were talking about protein production. Now this illustration is specific for our eukaryotic cell because there's a clear separation of the nucleus and the cytosol. So this is just a quick little summary. As you can see in the nucleus, we talked about the fact that we had to do our initiation, our elongation, and our termination, and use our template DNA strand to go ahead and produce our complementary RNA strand, which then had to be modified to make sure that it was nice and stable and would be able to be exported out of the nucleus. And the modification included the capping on the 5' end, the addition of the poly A tail on the 3' end, and obviously the splicing to get rid of the introns and go ahead and maintain the exon order. Once that was all done, we went into the cytosol, which obviously is the liquid within the cytoplasm, which is the area. And there we once again did initiation, elongation, and termination, but this time we did it in the name of translation. And our goal was to utilize our mRNA and read it in sets of 3, aka our codons, and the rRNA, which was composed of a small and a large subunit, was the one that was doing the reading, and the tRNA was the one that was bringing over the amino acids so that we could keep growing our chain. And once we reached our stop codon, the poly chain was released, and then we needed to do some modification to get it into its functioning form. And whenever it was no longer needed, we would degrade it, get rid of that protein, and then go ahead and repeat the steps all over again. That is the end of our chapter 7 discussion. I do want to encourage you to take a look at another PowerPoint that I have posted that is going to be an example of transcription and translation. And what we're going to do in that example is we're going to take a DNA sequence and we're going to run through the production of an RNA molecule. So that will be our transcription. And once we have our RNA molecule, we're going to go ahead and break it down into codons and use our codon table to figure out what protein sequence we'll produce. And that will be our translation. So whenever you're done with this, go ahead and make sure that you pull up the next file, which will be your chapter 7 transcription and translation example.